Good afternoon. I'm very pleased to welcome you to the session Nanophysics and Mathematics in Healthcare. My name is Inge Herrmann. I'm going to be chairing this session in the coming 1.5 hours. And I would like to strongly encourage you to ask questions via the interface so we can have a nice Q&A session. I would like to kick off this session by introducing the first speaker. The first presentation is going to be given by Dr. Yaroslav Nikolaev. He's a team leader of computational biology at InterAx Biotech in Villigen, Switzerland. Yaroslav, the floor is yours.
Good afternoon to everybody. My title slightly changed. So first of all, yes, I'm grateful to organizers for the opportunity to speak here. The title and contents of the talk slightly changed, but I think I'll try to show you even more interesting results which we get on combining artificial intelligence with the systems biology in GPCR drug discovery. First off, what are GPCRs? G-protein coupled receptors, if you don't know yet. These are the largest class of drug-targeted molecules at the moment. There are about 800 GPCR genes in human. This is the largest membrane protein family. And these are essentially one of the main sensors through which the cells perceive the world. 34% of all FDA-approved drugs are targeting GPCRs in the conditions like diabetes, cardiovascular, cancer, and brain disorders. And GPCR signaling combinatorially activates in the cell at least two downstream pathways, G proteins and aristines. And at least one of the G protein trimers gets activated. There are 16 G alpha main subunit variants, and with beta and gamma together, this forms about 1,000 possible combinations. Then there are also 3 types of aristines, and there can be different phosphorylation patterns on the GPCR in the intracellular part, in total giving about 10 to the 4 or more possible combinations of the signals this can generate. And this beautiful signaling complexity makes some challenges when you try to target these receptors with drugs. Namely, I can show this, illustrate on the enzyme inhibition drugs as an example. So cyclooxygenase is an enzyme which helps produce prostaglandins, and these molecules activate pain and inflammation in the body. And there are drugs which block it. You probably know aspirin and ibuprofen. When they block the enzyme, the prostaglandins are not synthesized and pain inflammation is reduced. So if you look at the enzyme basal activity, you can see that when you add the drug, it goes to 0. It's relatively simple in this setup, but it's not so easy for GPCRs. Most GPCRs have some basal signaling activity, and they are drugs which are called neutral antagonists, which force receptor to stay at this basal activation level no matter what. Then there are drugs which can shut down the signal. They're called inverse agonists. And there are also drugs like partial agonists and full agonists who can actually increase the signaling activity of the GPCR. And if this wasn't enough, actually GPCRs activate signals on different pathways, on several pathways at the same time. And then you can get quite a bunch of different responses you can get from one signal, from one molecule. And it's not always clear which of these signaling pathways will exactly be enacted when you look in vivo with the drug. And our company, InterAx, developed a unique technology which gives mechanistic insight about GPCR drug activation. And with this, we can better predict what responses these GPCR drugs will generate in vivo. And now we are further combining this technology with machine learning AI, which I will speak about shortly. So if Zoom shuts down, this is the presentation summary. We're at the verge of being able to predict in vivo effects of GPCR drugs from drug receptor structures, and systems biology with machine learning plays a crucial role here in between as a stepping stone to predict one from the other. Okay, and first some terminology. Beta-2-AR in the presentation refers to beta-2 adrenergic receptor. It's a showcase receptor used here, and it's a target for asthma treatment, among other things. Residence time refers to the time one molecule spends bound to another, and it's an inverse of kinetic dissociation constant. And ML refers to machine learning, a subfield of artificial intelligence, which is most common today applications. Many GPCR drug candidates fail during transition from in vitro studies to in vivo tests during drug development. And one of the main reasons for failures here is that there is a sharp increase in complexity in between the two. In here, you're working with isolated molecules and cells, and you characterize drugs with few parameters like KD and efficacy. And on the other side of these transitions, you are actually dealing with a complex system and hundreds of parameters network. And due to this sharp increase, it's hard to predict what's happening afterwards. And due to this poor predictability, there are high failure rates. Systems biology approach allows to alleviate or smoothen this transition by creating by recreating part of this in vivo complexity in silico. So in a bit more detail, how is this different to other approaches? The classical approach, you would take drugs and analyze them often with static assays like endpoint assays to figure out the desired phenotype. With this, you determine parameters like KD, potency, efficacy, and then based on this, you decide which drugs to test in vivo. It works well for enzyme type of drugs, but for GPCRs, it's a bit of a hit or miss because you really don't know what's happening on multiple different pathways that they can activate. At InterAx, we perform much more detailed characterization of GPCR drugs. We run kinetic assays to measure time-resolved responses in the cells. We do multiple assays at the same time. And more importantly, we integrate the data from these assays using mathematical models to see how these assays crosstalk between one another when activated by GPCR. And with this, we can extract a lot of mechanistic information and sort of like a fingerprint of the GPCR response. And we call this approach model-guided selection, which is based on multiple mechanistic parameters and allows us to better predict which drugs will work in vivo and why. And going into even more detail, we have about half a dozen different kinetic assays at the company, and in many cases, these two assays shown here provide the most crucial information. First off is the cyclic AMP signaling. When you activate the GPCR, in particular beta-2 adrenergic receptor, here with agonist, the cyclic AMP levels in the cell increase, and if you block the receptor afterwards with antagonists, they go down. And then second, most activated GPCRs are regulated by internalization into the cell, and we can monitor this process with fluorescent markers. And again, when you add an agonist to the receptor, you see internalization going up, and you add antagonist, it goes down. Receptor recycles back. And we do this for multiple concentrations and also usually for several different drug candidates in parallel. And then we can feed all this data simultaneously to an ODE-based mathematical model shown schematically here. And this model encapsulates all our knowledge about the target GPCR system. And the predictions of these models you can see here as thick lines. And this analysis yields a number of parameters, some of which I show here, like K-on and K-off of the ligand binding, K-on and K-off of the G-protein, and beta-arrestin. This yields kind of a fingerprint of the system, as I mentioned. And each of the drug candidates we test gets such a fingerprint characterization. And what's more important, our models can explain these dynamics really well, and this holistic analysis allows us to extract certain parameters like the residence times on K-off of the G-protein, which are simply not possible to measure experimentally at the moment with any approach. And connecting to what I shown before, these fingerprints kind of encapsulate the GPCR signaling patterns. The G-protein trace in the fingerprint encapsulates one of the pathways and beta-arrestin the second one. And there are a lot of interesting things you can do with analyzing these fingerprints when you have a big data set of drugs, and I show one example next. Yes, so in the data I show here, we work with this beta-2 adrenergic receptor, and it's a target for asthma treatment, and we analyze 20 compounds agonists which are used as a treatment of this condition. And in treatment of asthma, these drugs are often characterized by the duration of their action in the organism when you administer them. And these are classified as short-acting drugs, long-acting drugs, and ultra-long-acting drugs. And this duration of action is to a large extent correlated with compound lipophilicity, and this parameter was often used to kind of roughly categorize them. And you can see the short-acting drugs really stand out based on this parameter, but there is an overlap between long-acting and ultra-long-acting drugs. And we can try now to separate these classes even further by their in vivo effect if we resolve it in a second dimension using some mechanistic parameters. If we use a KD parameter of the ligand binding to the receptor, you can see the resolution map here, and you can see that the long and ultra-long acting compounds are still overlapped. If, in addition, instead of thermodynamic KD, you use kinetic K-off parameters, the time which ligand spends at the receptor, you can get better separation of these two classes, long and ultra-long acting. And if you further combine this ligand K-off with a K-off of the G-protein effector from the bottom of the receptor, you can actually get a really good separation of these in vivo acting drug classes. And you can start predicting the duration of the compounds based on the systems biology data. And we know from internal analysis of different receptors that this approach works for different receptors once you gather enough mechanistic information on one side, and you also have some in vivo information about the effects these molecules can generate. So in summary of this part, I've shown you that we are able to predict certain in vivo effects of the drugs based on systems biology reaction parameters. And only this by itself already gives quite a bit of a boost for the drug development pipeline. And now we are enhancing it even further using the structural biology with machine learning, which I will show next. When you have drug-target structures on the computational side, it's an often thing to do virtual screening to try to find good binders which can bind to your receptor with high affinity. We went that path and could predict some quite potent new agonists for the beta-2AR receptor, shown here as molecules 1, 2, 3. And you can see their cyclic AMP response in comparison of a standard agonist isoproterenol. However, since GPCRs are challenging targets, knowing their KD and potency in this setup is not enough to really predict how they will behave in vivo because of this problem of high complexity, which I mentioned before. So to generate more information and enable these in vivo predictions, you can use a model of intermediate complexity like the systems biology models that I shown before. And I will show you now how we are deriving and predicting systems biology parameters from the GPCR structures using machine learning and structural data. So the training set, to train the machine learning models, we use two data sets. One I already mentioned, we have a systems biology fingerprints for about 20 or more beta-2AR agonist molecules. And then we also have on the structural side, molecular dynamics simulations of beta-2 adrenergic receptor in complex with these molecules. And these molecular dynamics simulations, they last about 1 to 10 microseconds, and they capture dynamics of the receptor in presence of specific drug. And then we further train machine learning model to predict the kinetic parameters of systems biology data from the dynamics of the receptor. And once we have a few hundreds of these data sets, we would be able to predict whole fingerprints at the same time, but at the moment we have only a couple of dozens, and therefore we are predicting only individual constants step by step, which I illustrate now on the example of the ligand residence time. So we take all the ligand residence times in this data set, plotted here, and we split them into three classes, with high, medium, and low values of this parameter. And we train the machine learning model to predict these parameters based on the structures of the receptor. And there are different machine learning model types and setups and lots of hyperparameters you can train, the ways that you represent the data. So you need some validation. What's the best approach to do this? And here I show you the results from our neural network training with the validation, what is called leave one out approach, cross-validation. So in this setup, you take basically one of the compounds from a data set, you omit it in the training, then you train your model, and then you ask the model to predict to which of the classes this compound belongs to. And you do this for all of them, and this way you train the model. So here you can see the the compounds from these three initial classes, and if the compound dot is in the diagonal box, it means it was predicted correctly by our model, and if the compound is off diagonal, it means it was incorrectly predicted to a different class. And then we have some parameters which are on the edges of this separation. They're most often predicted incorrectly, and we don't include them into the into the true positives or false positives. So with this overall training, we can get at the moment about 65 to 75% prediction accuracy on this parameter, which is pretty good, I would say, knowing what other models in the field can achieve. But a reasonable question here is to ask, what about real world scenarios when you have some data in the production and you want to predict something for new compounds which the model hasn't seen? So I come back now to these three novel agonists that we predict before, which the model hasn't seen at all at this point, named here 1, 2, 3. And our model predicts that two of them will be in the medium koff.Ligand class, and the third one will be in the high class. So we do the experimental validation, we measure cyclic AMP and internalization assays I mentioned before. Here is the data for the third compound. Then we feed them with the mathematical models that we developed. We derive the fingerprints, including this koff parameter, and hooray, we could predict 2 out of 3 compounds correctly. And this is a pretty good result for one of the first trials that we made. And what is also highly encouraging is that we achieve a comparable accuracy on these novel compounds to what we achieve in the training set on the 20 ligands that we tested before. Okay, so this is with the ligand residence time. So the question is, can we predict other parameters like G-protein residence time? The G-protein here is the signaling protein which binds to the receptor in the intracellular part, and there is this parameter in the fingerprint G-protein koff. So we do a similar thing. We take this parameter, split it into 3 classes, and we try to predict which ligands belong to which. And here again, we were able to achieve with neural network training about 75% prediction accuracy. This is all really new data, so we don't have yet validation for the new compounds shown in red dots here for the koff G-protein parameter. But what brings us confidence that this is also holding true is the following. Our machine learning approach finds known GPCR structural motifs to influence this koff G-protein parameter. More specifically, we rediscovered 2 motifs called PIF motif and NPXY motif, shown here, to be influencing this parameter. And these motifs are highly conserved across all class A GPCRs and were described to correlate with certain aspects of GPCR function. We were quite amused to see this, to be honest, and in a way it's like we have now ultimate discovery kit in silico for finding GPCR structure activity relationships. And we are now actively exploring this trove of new results and data. So coming back to this slide, in a summary of this part, we can predict the residence time on koff of the ligand and G-protein using machine learning and structural data. And we expect that we'll be able soon to predict other parameters in these fingerprints using a similar approach. And speaking of these 2 constants, koff-L and koff-G, these are exactly the same constants which I showed in the first part that we could correlate with the in vivo action, duration of action of the beta-2AR drugs. So in a nutshell, we're becoming able to predict structure or in vivo effects from the structures. So we combine the machine learning with the systems biology, real intelligence to become able to design drugs, GPCR drugs, with specific in vivo effects. And with this, I would like to and presentation here. This is really a team effort, and I would especially like to thank Tomek, Georgi, and Agni on the computational side for their hard work here, and also Joachim, Vikram, Miriam, Nadine, and Alessandro in the lab, and also our collaborators. And as well, I would like to say many thanks to European Union Horizon 2020 program, through which a big fraction of this project was supported. And thank you for your attention.
Thank you very much, Yaroslav, for a beautiful talk. We have a question from the audience. Miriam is asking, can multiple agonists act on one GPCR? If yes, is the activation effect additive? Or can actually two partial agonists lead to reverse agonist action?
It's a bit tricky. I would have to internalize. So in two questions. So it depends on the GPCR. Usually, or for many of them, there is only one binding pocket. So essentially, you can only choose like which one agonist you bind. For some GPCRs, you have also allosteric pockets, and then you can bind two molecules. And then maybe these effects could start being additive. And yes, if this partially, yes, and then there could also be things like one ligand could start blocking the exit of another one from the receptor. So you can think of these combinations and try to figure out whether you can combine them properly or not. But there is another dimension when two GPCRs can bind into a dimer, and then there is even more crosstalk between them. But this is a bit tricky, and people are still trying to figure out and learn more about this.
Thank you very much for this very clear answer. Unfortunately, we already have to move to the next presentation that will be given by Dr. Marianne Marchioni. She will be talking about single particle measurements of number, size, and charge that are required for confidence in nanomedicine engineering and development. Marianne is an application specialist at ISON Science in Lyon, France. Marianne, the screen is yours.
Yeah, thank you very much. Is it okay? Can you see my screen? Okay, perfect. So good afternoon, everyone. My name is Marianne Marchioni, and I'm the application scientist at ISON for the European country. And today I will talk about how a standardized single particle measurement of number, size, and charge is required for confidence in nanomedicine engineering and development. So today, if we are looking at PubMed, for example, for nanomedicine or nanobiology, we can find 100,000 of publication. But some of the publication shows results that are not reproducible. And this is a waste of time and energy. And this is mostly due to the technique used for the characterization of the particles themselves in the studies. And we all know that good size measurement is essential because the biological and the chemical activity of this particle are directly related to the size of the particle. And measuring the size could be quite easy depending on the instrument you have. We also know that the particle concentration measurement is an essential part of the data sets, especially if you want to describe totally your sample, or if you want to normalize the further investigation. And this is why a high precision single particle size, charge, and concentration measurement is a handy tool to have in a lab. For example, I'm showing you right now the result of a comparison study where the most used technique in the nanofield are used to analyze a trimodal suspension made of polystyrene beads. Here we will talk about the DLS, the TRPS. This is the device from ISON, the NTA, and the DCS. And in these results, it looks obvious that the optical technique cannot resolve the sample complexity. Only TRPS, as a real particle-by-particle number-based distribution, provide a realistic and accurate measure of the particle size distribution. Here you can see the three subpopulations of the sample. So how this device is working. Everything is based on the Coulter principle. We are using a nanopore, this cross made of plastic, where a tiny hole is made at the center of the pore. And then we are applying a voltage across this nanopore to create an ionic current. And then the particle will go through the nanopore one by one and create a blockade of this current. And this blockade is really rich in information. The magnitude of the blockade will give us information about the size of the particle. The duration of the blockade will give us information about the charge of the particle. And the number of blockade will give us information about the concentration of our sample. And then we will calibrate each measurement with a very well-known sample made of calibration speeds in the exact same condition as the sample itself. The same buffer, the same pore, the same voltage, the same pressure. This way, results of TRPS will give you exactly what you have in your sample in the size range of measurements. TRPS is not fitted to a model. There is no assumption in the results of a TRPS measurement. And this kind of measurement leads to this kind of results. Here you can see the three subpopulations, the size distribution of each of them and the mean size of each of them. Also, you can know the concentration of each subpopulation as well. If you want to go further in the characterization of the particle, we know that the zeta potential is really important to understand the composition of the surface of the particle. The TRPS, as a single particle analysis device, can analyze the charge of each particle one by one and give you the results of the charge of each particle in function of their size. This way, you can discriminate in the same sample two subpopulations of particles depending on their zeta potential. For example, here, the blue population and the red population, both of these populations are made of particles of 100 nanometer diameter. But the blue population, are more negatively charged than the red one. Or another example, the purple and the light blue population. They are both made of the same particle, but the purple one are covered by DNA. This will affect the zeta potential, and this way you can discriminate the raw particle, the crude one, from the coated one. So in science and in research, complexity is the norm, and TRPS will give you the higher resolution in multiplex analysis, giving you simultaneous the determination of the size, the concentration, and the zeta potential of a given sample within a given size range. At the top right of the screen, you can see the view of the ongoing measurement of a quadrumodal suspension of particles. Here you can see 4 lines made of red dots. Each dot is a particle who goes through the pore. This one are the smallest particle, this one are the biggest particle, and each dot is related to a blockade. You can see here you can see the big one, here you can see the small one. At bottom right here, there is a view of the ongoing measurement of a sample made of extracellular vesicles. You can do this kind of measurement for particles from 40 nanometer to 15 micrometer. So it allows you a broad range of measurement. So, in summary, TRPS will give you information based on the biophysical property of particle itself. It allows for meaningful comparison between the concentration over a size range of interest and the concentration of the total sample. For example, if you are working with extracellular vesicle in a biological sample, you can know the concentration of the extracellular vesicle themselves and know the concentration of the particle of the full sample, for example, extracellular vesicle, apoptotic body, and the protein aggregates. When I'm talking about biophysical property of the particle, I'm talking about the size distribution, the particle concentration, and the surface property, so the charge measurement. And these properties are used in nanomedicine to do biomarker discovery, diagnostic, biosensing, or in therapeutic to know more about the percentage of drug loading or to the release profile of drugs. Until now, to perform this TRPS measurement, the Q-Nano is or was the gold standard, but the device has to be handled and tuned manually, and this can be quite challenging for the beginner user to use the device. And this is why we have worked and developed a fully automated device, the Exoid instrument. And this way, it reduced the variability between an experienced user and a beginner one. And the technological advancements have reduced the noise of the measurement and allow now the measure of particles below 40 nanometer. In nanobiology, we know how difficult it is to isolate the particle of interest from the media. And we want to propose here fully standardized method to isolate the particle from the media. It's based on size exclusion chromatography, and it's available from samples from 100 microliter to 100 milliliter. It will remove 99% of the contaminating background protein, and the more important thing, it will maintain the structural and integrity and biological function of the particle to allow you the better results in the further investigation with your particles. So, as a conclusion, I show you right now a plan of experience you can do working in the extracellular physical field, for example, using the ISON products. First, you choose your biological field. It could be plasma, urine, blood, serum, whatever, and choose the suitable column for your sample. Then do the collection of the fraction. If you are using an automatic fraction collector like the IFC, it will ensure you the more reliability and the more reproducibility in your isolation process, and it will allow you to do something more interesting than counting droplet in front of a column. Then select the fraction of interest, the one containing the particle, and perform TRPS analysis to know the size distribution, the concentration, and the charge of your sample. Because TRPS analysis need only 35 microliter of a sample, you can use the same sample to do Western blot analysis, proteomic, lipidomics, or other downstream analysis like RNA-seq using the QEV RNA extraction kit. And at the end, you can use the TRPS analysis to conclude your measurement. So the research and the science worldwide need for reproducibility. And reproducibility requires standardization of the method. It starts with the sample preparation and purification and ends with the analysis of your sample. Most methods will provide significant variation in results, even if the measurements are accurate. The size exclusion column, the QEV, offer a very simple and efficient and standardizable good solution for purification. The TRPS, being always calibrated, also offer a simple and effective method for the standardization of the characterization. And to perform a full standardized journey in your analysis, you can use the QEV RNA extraction kit to keep the standardization from the beginning to the end of a downstream analysis. Thank you very much for your attention, and I will be glad to answer your question.
Thank you very much, Marian, for this clear presentation. At the moment, we don't have a question from the audience, but I would just quickly like to know, are there any disadvantages or downsides? Because it looks like you found a magic technique.
Can you repeat your question, please? Because I didn't understand all the word.
Can you just quickly say if there are any disadvantages of your technique? Because it sounds really magic.
Magic. Oh, disadvantage. Yeah. So first, here I thought you cannot analyze particle below 30 nanometer. So for now, we are working on it to improve it. But for now, all the particles below 30, we cannot see them. So that's the first disadvantage and the main one.
Thank you very much. So in the interest of time, we're going to move to the next presentation. That's going to be given by Professor Gabriel Apley. He's the head of photon science at the Coulter Institute in Villigen. He's a professor of physics at ETH Zurich and at EPFL. And he's going to give a presentation entitled Combining Existing and Novel Developments for Sustainable Medical Solutions. Professor Apley, the screen is yours.
So I'm not sure I'm in the right session here because at least my abstract doesn't seem to fit in the session, but since there's a few people still here this late afternoon, for which I'm grateful, I'll just talk you through a little bit about what we're doing. And so I'm representing a company called Aquafirm. And Aquafirm is a multidisciplinary London-based company with the CEO is David Sarfy, who was involved with the original powder jet venture, which was later sold to Pfizer. John Wood, CVE, who's chairman of the board. He's now mainly associated with CERN. A very important person, Dr. Rob Merrifield, who's director of software. Myself, my main history is at IBM, NEC, and AT&T. And Tony Cass at Imperial College London. And what we've built is a technology that links sensors or software and particularly mapping software to track pathogens and disease. And I'm just going to take you through some of the products that we provide to people. So let me just see here we are. So our main backbone is actually a digital platform. And that digital platform essentially has access to all kinds of inputs, particularly sensor inputs. You can see here the last point, we're basically having water sensors. Of course, these days water sensors could also be biosensors. For example, they could be testing for the presence of viral matter or antibodies. And then once we have such a sensor network, of course, that sensor network can be input via usually the mobile phone network to the actual software platform. The software platform allows visualization, but it also allows planning and optimization. And one of our actually clients that we have right now in Brazil, their main interest is actually in optimizing a greywater network. You may realize, of course, that there are different quality of water that could be used, for example, for gardening or for showering as opposed to drinking. And in many countries, this is not so true of Europe and the US, but many less developed countries, there's a push to separate those networks to reduce cost and in fact also to reduce environmental impact. And so we found ourselves actually there a niche in supplying software for optimization of greywater. And basically this gives us this possibility to plan water infrastructure, map and optimize routing, and also calculate sort of capital expenditure and operating expenditures for these types of networks. And this just shows sort of how the software works. What we really have, though, as the core, and this should really be more interesting to you than the particular application I just spoke about, is we have the core of this GIS-enabled mapping. Now, GIS-enabled mapping means that if you have a biosensor, for example, the type that we were hearing about in the previous talks, and that biosensor could be essentially plugged into a mobile phone, which, as you know, is a GPS chip. That mobile phone, the location of that mobile phone is known, and then that can be put into mapping software where we have all kinds of other maps, for example, water, railways, protected areas, elevation, roads, and so on. And then we can make correlations. So if, for example, we're looking for disease clusters, which are measured, let's say, using one of your fancy biosensors, then people, physicians or nurses in various testing stations here could be inputting those data directly via basically their mobile phones. And then we could build up a correlated database where we could, for example, relate, let's say, water to particular types of illnesses, roads to other types of disease, elevation, let's say, to health. All of these things are basically within the capability of our package to analyze. And then, of course, once you've analyzed the data, we can perform optimizations. In this case, we're optimizing a route of a water line. This is actually the solution that we have to route water from this location, clean water from this location to that location. But we could have just as well optimized to determine the boundary of, let's say, a zone where we imposed quarantine with respect to some disease. Where we said, well, people who live over here had better stay home all the time. The people over here can be a little bit more free to travel. Now, we actually are not just a software company, we are a biosensor company. And in fact, we started out, Acknowledgements started out as a biosensor company. We realized actually at the end of the day that biosensing, particularly when it has to do with large populations, is really a software problem. So we added a software team. And actually the way the company started out is that we developed a synthetic biology route to rapidly test for arsenic in drinking water. This is a very serious problem. It affects 140 million people in 70 countries. Huge numbers of people die from this every year. Actually, the scale of the numbers is just as bad as the scale of the numbers that we're seeing with COVID-19. Bangladesh is a particularly acute problem, and this is just a horrible picture of what happens when you get arsenic poisoning. Basically, what happens to you before you actually die. And what we've developed, we've packaged up this biosensor, and basically it's a patented sort of test strip-based sensor, very much analogous to a pregnancy test. It's rapid, you get results in 3 minutes, with training, no on-site supervision. And then it's web connected, it's a data upload, so as more and more people use it, we have a network advantage in actually defining the problem from the public health point of view. Currently, actually, the current tests are terrible. They're complex, they're slow. Actually, many of them are in fact dangerous because they involve mixing up chemicals in the field. And strangely enough, or not so strangely, there's no web connectivity. So when people do tests, they may know what to do on their own, but they will not be able to really provide useful information to further policy steps. I should say that this software, of course, once we have the software, we have the sensors, we have the readers, we have the smartphone phones, this data goes into the cloud, and then suddenly, when we collect enough data from enough towns, in this case this is Bangladesh, from enough wells, we can start getting basically real-time mapping and data analysis. So actually, you have to have field tests in Bangladesh. This actually has been the topic of a video by the BBC. I don't know how much more time I have, but I would love to show you the video if there were enough time. We've basically demonstrated very clear benefits of the synthetic biology test over the sort of the conventional chemical tests that are out in the field today. So how we really work with customers is we essentially have the full suite. We have sensors, but we combine the sensors with software for basically monitoring contaminants in water. And of course, this deployment and this software product that we have provides, of course, a base as well for our consultancy business. But the main business we're really in is the digital business, exploiting sensors with web connectivity. I'm just going to remind you of the CEO of the company, because that's the person who's really managing the whole thing. I'm just a non-executive co-founder of this enterprise. That's his email, and then this is the web address. Now, of course, during the COVID-19 pandemic, of course, we've put our skills to use for problems that are also facing rich people rather than just poor people who have arsenic in their drinking water. We have actually, if you go online, you can actually find out where you could travel to from, for instance, Switzerland. We have actually a map of the world which actually displays sort of prevalences of the disease in cases per 100,000. Switzerland is not in a very good shape at the moment. And by clicking on the country, then you can actually discover the number of the travel restrictions. In this case, this is traveling from the UK.
Could you wrap up in 2 minutes? Could you wrap up in 2 minutes, please? Thank you.
So this gives you a taste of the type of product we have. Here we're again interfacing a mapping software, in this case with detailed information from all kinds of government websites throughout the world. So thank you very much. How much more time do I actually have?
Not that much, but we can have a Q&A session at 4:45, and
okay, great. So
that's time for more questions.
You can go and visit the websites, acrofirm.com and chronotravel.world. COVIDsim.org is a simulation site where you can actually play policymaker and see, again, use the computer software that we've developed to actually predict what the outcomes would be of various policy steps you might be taking in response to COVID-19. Thank you very much. Thank you very much
For this presentation. Unfortunately, we have to move on with the program already now. So in the next 10 minutes or so, I will give you a little overview on what we've been trying to do in a quite different field. So we work on inorganic nanoparticles, and we've been trying to rationalize the design of these nanoparticles by taking different approaches. My group is really interested in removing pathogens from blood in a process that we call magnetic blood purification. And if you look at the blood, there are many, many cells, like we have about 5 million red blood cells in a microliter of blood. And then occasionally during disease, we have also pathogens, but they're much less abundant. So we have like typically 10 pathogens in that 1 microliter of blood. And then we have a couple of phagocytes, like neutrophils or immune cells, and their task is actually to keep the bloodstream clean. So what we would like to do in our process is we would like to selectively remove these pathogens, but of course, like no other cells. And we would do that in a process shown here. So similar to diagnostics, where magnetic particles are used to selectively remove toxins or specific cells from complex samples, we would do the same here, but in a continuous extracorporeal manner, where we would take the blood from the patient out into a extracorporeal loop, similar to dialysis. We would inject functionalized nanoparticles that would bind to the pathogen and would then be removed magnetically. And the clean blood would be recirculated. A hallmark of this process is that the time you have to complete this process is very, very short. It's typically a minute or so. And the challenge really is that whereas for small molecules that are typically removed by classical dialysis, small molecules move very, very rapidly in the fluid, and they can readily bind to even like stationary immobilized adsorbents within a reasonable time. However, if you look at large pathogens, they don't move as quickly in the fluid, and they're rather stationary. So it takes them a very, very long time to adsorb on a stationary filter or surface. So our approach was to take small nanoparticles that can screen this fluid volume in 3D in very, very short times. And we were interested if in this way we can also efficiently remove larger pathogens in very, very short time frames. However, this is a very complicated process. And in order to understand a little bit better on how many particles we need to add in order to remove how many cells, and also how selective our binding event needs to be, we built a model based on population balance equations, where we can look at the number concentration of unbound beads, and we can look at different cell populations and the number of beads attached to them. So typically we have, for example, 2 cell populations, in orange the target cells, and in green the non-target cells. And we can look at how these bead concentrations immobilized on these cells evolve. It's important to know that, of course, the magnetic removal efficiency depends on the number of beads attached to these cells, and we will only remove cells that have sufficient beads attached. It's it's very hard to explain it in 10 minutes, but what we can essentially play with is we can play with the ratio or the concentrations of these two cell populations, and we can play with the selectivity of the binding. And that we can see here in this graph on top that if the binding is very selective, we remove all the target cells, but none of the non-target cells, the phagocytes. However, if the binding is not selective anymore, we basically remove all the cells. And this is, of course, not what we want. So we use this model to optimize the bead concentration and the incubation times. And we could show that indeed we can, for example, very selectively remove, in a model system, of course, circulating tumor cells, indicated here with very high separation efficiencies, with functionalized magnetic nanoparticles, whereas there was very little interaction with human monocytes. And then we can also optimize, like this is now verified experimentally, we can optimize the size of the beads. Beads that are too small are not efficiently removed by the magnetic separation process, and beads that are too large, they are just too slow to bind to the target cells. And of course, the entire process is strongly concentration dependent, which is actually nicely also seen in our model system. What we also could see in our model was that there is this competitive uptake or binding to neutrophils. These are like non-target cells, and this is strongly time dependent. And we could also verify that experimentally, where you can see that if the contact time between the beads and the blood is too long, you lose neutrophils quite quickly. And we can also visualize this in transmission electron microscopy, where you could see that, in fact, tumor cells, which are target cells, carry like these magnetic beads on their surface bound to Epcam receptors, for example. So this works really nice. However, if you incubate for too long, you can also see that these particles get internalized by phagocytes. And then once these phagocytes carry enough particles inside, they're actually also magnetically separated. And this is what you see here. In the second part of my presentation, I would just briefly like to show another example how we try to rationalize our nanoparticles design by taking a slightly different approach. So in a project where we try to develop a new generation of tissue adhesives, we saw that we can maybe tailor the activity of these particles by tailoring their architecture. So we wanted to address the phases of wound healing and develop particles that can optimally in a spatial temporally way address the phases of wound healing. And we literally constructed a particle system, a core shell particle system that can, in a temporally orchestrated way, optimally support these phases of wound healing. So first we can have coagulation, and then we can have like anti-inflammatory activity, antimicrobial activity, and some tissue regenerative activity, all in a temporally orchestrated way. This is a particle that at the moment we call nanoglue. We took an approach to really combine different materials that were known and were described in the literature in a way that we thought would make sense from an architectural point of view. So we selected two materials here and sprayed them in a flame spray pyrolysis process where the particles can be synthesized in a continuous way in a defined architecture. And we could really verify that the particles really have the properties we wanted them to have. So for example, they show very strong tissue adhesion for bioglass containing particles, whereas for the other like ceria particles, this adhesion is much lower. And if we can generate a hybrid system that actually shows the same adhesion as bioglass, which is much stronger than the surgical gold standard. So we can have strong tissue adhesion, and then we can also achieve blood clotting. So if we have here like just the control, the negative control in PBS, we can see that we can achieve faster blood clotting with this nanoglue which is highly desirable in this wound care setting. Additionally, and now comes the serum part, the serum oxide part. These particles can protect cells from oxidative stress. So if we look at the cell survival here, and in cells that were stressed by hydrogen peroxide, which simulates a wound environment, we can see that in the untreated cells, all the cells die, whereas in the particle-treated cells, a significant portion of cells survive. So we have a cytoprotective activity, and additionally, this nanoglue also shows antimicrobial efficiency. Of course, we want to see if this really works also in vivo. So we went to our clinical collaborators, and they actually confirmed these promising properties for these nanoparticles in an in vivo model, where they transplanted a skin flap to the back of a rat. And we looked at the tissue regenerative activity. So we looked at the blood flow in this flap that was significantly increased compared to clinical gold standard. This could be confirmed that the particles actually promote the formation of blood vessels. Here I'm showing a tube formation assay, and they have similar activity as the well-known positive control vascular endothelial growth factor. And this improved blood supply actually also results in an improved flap survival compared to the clinical gold standard. This was highly promising, but we also wanted to understand more and learn more of what these particles do after application. And we developed a process to learn from that and redesign our particles. So we tracked our particles and we tried to refine them in vivo. So we took a skin biopsy sample and we measured computer tomography, CT. So you can here see dense parts which represent particles. So this helped us to find the particles. I'm just going to skip that. And then we can cut the skin biopsy and into a histological section. I'm showing here the cross section now. And we can look at the distribution of serum by X-ray fluorescence in this tissue sample. And high concentrations are now shown here in green. So we know that most probably the particles are located in this region. It's actually a CD68 staining that also shows the macrophages here in brown. And we can see that the particles seem to co-localize quite well with the macrophages. We can then also confirm that this is not just serum, that this is actually seria in measuring top sims of the same region. And in this sense, we can look at the location of particles in histological section. So with this, I would just like to come to the conclusion that in situ analytics can provide really, really important insights to guide materials design and development. And we can learn from findings after application, where we look at how particles change after their application. Additionally, the integration of even really, really simple simulations to reduce the design space is an incredibly powerful tool. So with this, I would like to thank my group, the funding, and I would like to thank you very much for your attention. Perfect. So I think we're gonna move on straight with the next presentation given by Dr. Sara Fortuna. She's the coordinator of the Self-Assembly Recognition and Applications Group at the Department of Chemical and Pharmaceutical Science at the University of Trieste in Italy. And she's gonna talk about a computational protocol for the in silico maturation of antibody fragments. We're excited to hear your presentation.
Thank you very much. And thank you all for being here today. So my name is Sara Fortuna. I lead the Self-Assembly Recognition and Application Research Team. And our mission is to provide novel solutions to problems of medical interest through the computational design of new molecular systems. We are a group of computationals. And in particular, we want to answer the call of an increasing, And in particular, we want to answer the call of an antibody market calling for new ligands for therapy, diagnostic, and research. We know that the process of antibody discovery can be a long and tedious process. And even if the pace of antibody discovery steadily increased as new technology became available, only in this new century, the in silico discovery is becoming a real opportunity. This is especially due thanks to the availability of high structural data, cheap sequencing technique, and of course, powerful computers. In particular, in 2007, NVIDIA introduced video cards that could be used for scientific computing. And this was an important milestone for scientific computing because it allowed us to go from the simulation of roughly small systems to that of very large systems. The availability of powerful computer is coupled with the availability of excellent software developed in the past century. First of all, we have homology modeling code that allow us to design proteins when their three-dimensional structure is not available. We have a docking code that allows us to create complexes between a protein and other proteins, or proteins and small molecules. We've got scoring functions that could score and quantify in a very fast way the binding affinity between these objects that comes together. We've got reliable molecular dynamics techniques that allow us to follow the time behavior of our system. And lastly, we got new artificial intelligence code that are being designed that can let our system evolve. Of course, we have also many other software, and we want to build up on that. How? Well, we can link our software together, or we can mix them in a way that it pleases us, and it allows us to reach our goal. In the particular case of my team, our current main goal is to develop novel binders capable of capturing with high affinity protein biomarkers for the development of new ligands for medical, biological, and pharmaceutical applications. And in particular, here on the right, I show you small antibody fragments known as nanobodies. It's a single domain antibody of camelid origin. And that's the object we are looking at today. So we want to optimize this object, in particular its binding affinity, and to do that, we'll take advantage of Bind Designer. Bind Designer is a code that we use, recently developed, that link together state-of-the-art computational techniques such as molecular dynamics, scoring function, Monte Carlo-based optimization, and of course, side chains reconstructions. That's this code is an evolutionary code, and the brilliant thing is that it takes as an input only an initial complex that can be obtained either from crystallographic data or can be constructed by homology, modeling, and docking. So the code first appeared in 2012 thanks to the effort of the Alessandro Laio group in CISA, and then developed this evolutionary code in order to optimize peptides for drug recognition. The latest achievement was last year in 2019, where we used the same code together with Laio group to improve the binding affinity of nanobodies with the goal of recognizing protein biomarkers. So what we are talking about is the computational evolution of a biological object. Please note that we are not mutating the genome of the object, but we are mutating directly the proteome. This means that we mutate directly its amino acid chain or its amino acid sequence. As a model system, as an initial system, we took HER2. HER2 is a transmembrane protein and is a protein biomarker and is known to be associated with breast cancer. And our goal was to design or to maturate nanobodies capable of capturing this protein. And in particular, we want to look at the interface between the object we want to optimize and our target. On this interface, we see this highlighted in red, the CDR2, which is the area of the protein that we wish to optimize. How to do that? Well, we simply take our nanobody, we select the peptide chain here in red that we want to optimize, and we take a reduced representation of the protein, mainly only its binding domain, which, by the way, is the same binding domain of trastuzumab. The code then is fairly straightforward. In the code, we pick an amino acid at random, we mutate it, we estimate the binding affinity between the mutants and the target, and we decide whether to accept or not the mutation, and we repeat the process iteratively. At the end of our optimization, we obtain a curve like the one on the right, in which the predicted binding affinity of the nanobody reach a minimum along the simulated time. Of course, like in all vitro screening techniques, we generate a large number of sequences. With this optimization algorithm, we can evolve between 50 and 200 sequences. We pick the optimum sequences and we send 20 or 50 of those sequences to a further in silico screening based on molecular dynamics. And from this pool, we choose 5 to 10 sequences to be expressed in bacterial host. Of course, some of these sequences will not perform nicely, but in general, we can obtain 2 to 5 candidates which are good enough to be fully characterized, with the hope to obtain one lead. In particular, out of this optimization run, we took a nanobody called 44, which is our optimized nanobody, and we compare it to the initial nanobody called D9. So the first brilliant things that made us very happy was that actually the computational design nanobody was produced by bacteria. So it was possible to produce it in real cells. It was possible to purify it, and here I show you the size exclusion chromatography profile of both the optimized nanobody 44 in comparison to the initial hit or the wild-type nanobody D9. The optimized initial hit in this case was spun from a library. The second goal that we reached with this project, published here in Chemical Communications, was that the optimized nanobody 44 was shown by SPR to bind 10 times better its target with respect to the wild-type D9. And this is a brilliant achievement. We have shown that by optimizing a single CDR of an antibody fragment obtained by premium libraries, it was possible to maturate it and increase by 1 order of magnitude its binding affinity. So the second goal that we are trying to reach right now, we are working on this topic, is to answer the question, can we optimize a random binder? Or an alternative word, can we perform an ex novo optimization of an antibody fragment? Here in this animation, we can see again our nanobody, its target, and we can see at each simulation step, one amino acid of the CDRs is picked up at random, highlighted by a green van der Waals sphere, and mutated. And once again, mutation by mutation, we aim to reach a predicted optimum binding affinity. Again, we generated a number of sequences, screened the molecular dynamics simulation, and we gave a few sequences to our biologist. And what we obtained also in this case was that, yes, once again, we could produce in bacterial host our in silico design nanobody. And here you can see the sec profile, which has 2 peaks, not optimum. However, by sampling the larger peak, the gel looks quite pretty, and we could test it by ELISA, and we could show that actually it was selected toward one specific isoform of the biomarker we were looking at. So overall, in our team, we are part of the race toward automatization, and we take advantage of increasing computing power and increasing accurate software to design new antibody fragment. I showed you that we can optimize an initial hit, we can optimize a random binder, we can choose the binding site we wish to target, and we can produce these in silico designed antibody fragments in bacterial host. For instance, in the picture on the right, you can see our GFP tagged nanobody that they look really green and they are so pretty. So what next? Well, in the next future, we want to optimize our process, and really one very determining step is that we really need better experimental data to develop better and faster binding affinity predictors. So this I call for our experimentalist colleagues to take an action in this direction. Another things we would like to do in the next future, but actually our colleagues in IIT are already working on that, is to optimize a full antibody, so not only an antibody fragment. And then of course we need many new collaborators to tackle many new problems. To conclude, I would like to acknowledge all our collaborators, in particular Daniela Cesselli, that is a medical doctor that suggested us nice systems to work at, Ario De Marco, who is located in Nova Gorica and he's the expert in nanobody design, Miguel Soler, main developer of the code, and all the other collaborators of this project. I would like to acknowledge also my team, and in particular Barbara Medagli, who expressed in bacterial host all the nanobody we computationally designed. I thank the funding agency, in particularly the Italian Association for Cancer Research that is funding our work, and the Alternative Research and Development Foundation. Also, the code by Designer is implemented in a bash code and can be freely downloaded from GitHub, as well as its Python implementation called Parse. For this, please, you can get in touch with Miguel Soler, that is one of the main developers of the code. All our information, and if you want to know more about what we are working at, please visit our webpage at www.sarafortuna.eu. And to conclude, I would like to thank you all very much for your attention.
Great, thank you very much, Sarah. We unfortunately have to move on already to the next step, but we can have a Q&A session later on in the separate session room. Great, thanks. So the next presentation will be given by Dr. Dylan Yaroslav. He's working at the Department of Engineering at the University of Massachusetts in Boston, the US. And he's going to present paper-based immunoassays for surface chemistry and machine learning needs. We're excited to hear your talk.
Thank you very much, Inge. And thank you very much to the previous speaker as well, because it makes my job a little bit easier. So much talk about antibodies. And I'm just briefly going to show you how we use computational methods and how we use machine learning to improve the immunoassays which we make, mostly for contagious disease detection. You can see my presentation. Yeah. Okay. So the advantage of having this kind of immunoassays, paper-based immunoassays have a lot of advantages. And usually I would go into a very large introduction section where we talk about what kind of advantages, where they come from, etc. But I feel like 2020 has really introduced us all to the basics. So I'm just going to briefly use one slide to make sure everybody's on the same page. And the major advantages of immunoassays, of paper-based immunoassays specifically, is that they're rapid, they're cheap, they're very flexible in terms of design and use. They don't need power, they don't need any external devices, and they're very user-friendly. And in fact, they're so user-friendly that the readout can be 2 lines yes, 1 line no, very simple. And the signal on the line comes from what we call a sandwich formation. So you have an antibody immobilized on the paper, and then that binds to the antigen, and then that binds to the immunoprobe that we use. So immunoprobe is an antibody conjugated onto a nanoparticle in some way. And that's where the signal comes from, the color of the nanoparticle. In the most fundamental principle, the way that these tests work is that the antigen solution is dropped onto the sample pad, and then it diffuses through the conjugation pad where the antibodies on the immunoprobes bind to the antigens, and then they go, and if this has successfully happened, they bind to the test line, then the immunoprobes themselves bind to the control line, indicating that the test is working and there is flow, and then they possibly continue on what is in the test. Okay, and this is just to emphasize the flexibility of the whole testing system. Most people would be familiar with the fixed test where you have 1 line, 2 lines, and you make it in a factory and you send it off and somewhere, and people just generally use it for the one purpose that it is. However, nowadays, we may not want to do that. And there was very interesting talks in the previous days talking about mobile clinics and about testing at location. And in that case, you may want to have somebody who has more control over what the test is and wants to test for several things at the same time, for example, in one place, and then, or one patient, and then other tests for another patient. So to enable this, we've used the flexibility in collaboration with the Little Devices Lab in MIT to make a customizable sort of Lego-like test that you can build yourself. So you can make all sorts of funny arrangements like you see here. And you still maintain the cheap to make, user-friendly, etc. benefits. And I get asked by students all the time, like, why, if these tests are so great, why are they not out there? Why are we not using them all the time? And again, 2020 has been a great teacher on this. And the schematic of the test, the principles are very simple. You have 2 antibodies which both bind to an antigen and you see signal. Right, very simple. However, that's not the reality. In reality, these tests are subject to a variety of phenomena which we need to solve. And generally, we can summarize these challenges under 2 titles. One is sensitivity and one is selectivity. So the number of false positives and false negatives that these tests experience. And we can generally use 2 approaches to do this. One is the design-focused approach, which we usually, in our group, we usually do first. And then we have a more technologically focused approach. In design-focused approach, we talk about which antibody to bind, which nanoparticle to use, that sort of thing. And in the technological focused approach, we use advanced image analysis and data analysis and data acquisition and what kinds of data and machine learning, that kind of thing. And I'm going to just go through the process that we usually go through and give you an example of how this works. This is just one of our recent papers, and what we did here is we looked at an aspect which is commonly overlooked for these kinds of tests, which is the stability of the particles inside of a test itself. Now, it's often overlooked for obvious reasons. It's hard to we synthesized the number of immunoprobes with varying stability. And then we tested where they go in the paper and we tested their performance. And what we found is that particles which aggregate very a lot, they may not even enter the paper. And you get pretty much no signal. The particles which aggregate sort of a little bit, so this would be A is a lot, C in the image on the right is a little bit. This is, I feel, where most particles are when you make them. And the problem there is that they are relatively stable, but sometimes can aggregate. So small changes in the synthesis or in the running conditions can lead to variability in the test and can lead to false positives or false negatives. And then a particle which is very stable in size pretty much gives you a good reproducible positive signal. And that's what you see on the green graph here is that kind of good signal. So after we have the stability and all the tests, optimize, we can start looking at more advanced applications, and this is where machine learning comes in. So for example, we often have diseases which circulate in the same patient pool at the same time. And treatment is, however, dependent on which disease you have, and if symptomology is very similar, then that presents a problem. And we can solve that with rapid tests by utilizing cross-reactivity and by utilizing smart test design. And in this case, what we have is we have one strip, one test with 2 locations and 2 immunoprobes. So you have an anti-dengue location and anti-dengue immunoprobe, which is red, and an anti-Zika location and anti-Zika immunoprobe, which is blue. And what you get is that because the antibodies we use are cross-reactive, so they're not very high affinity with the antigen, but they do bind to the other antigens, depending on the pattern of binding and depending on the color of the spot, we can then use machine learning to train an algorithm and use component analysis to identify which disease. And as you can see, we identify the disease pretty well. So 3 out of 3 times we can identify the disease pretty well. And in more advanced uses of this approach, we can take a mixture of these 2 and use image analysis and discombobulate the image, and then use pattern analysis to, again, identify what's in there. Further use that we sometimes make of machine learning is we often employ SARS reporters because we use gold particles. And gold particles are some of the most commonly used particles for labeling in this. What we do is we use SARS reporters, and then that can help improve the limit of detection of these tests. And then we can use machine learning to identify and analyze the spectra so that the user does not need to understand Raman or other techniques to analyze the spectra. And this can also be quite beneficial for enhanced multiplexing, where we can do many different SARS reagents and SARS reporters, and we can have all of them identified, even in a mixture. And that can lead to a lot of multiplexing capabilities, even though the image itself looks the same. And so that's what we usually do for enhancing of the tests themselves. And finally, when we have the test working and we think everything's great in the lab, what we do is we usually field test them. So we send them to partners around the world in various locations, and when there is an outbreak. For example, we were working in the Zika outbreak in 2016, and we're working on the COVID outbreak at the moment. And we send both types, sometimes one, sometimes the other, of testing. And then we work with our partners to have those tests work for them and develop those tests with them. And that way we're trying to enable local diagnosis production. And because these tests are very simple and easy to produce, a lot of time you don't need a big factory. In fact, you only need maybe 1 room's worth of equipment. They can be produced at the spot. And what that can do is we can go around the usual problem with these kinds of epidemics and pandemics, which is stockpiling. And we can improve patient health outcomes in that way. And by monitoring and enabling local diagnosis and testing.
Can you conclude in a minute?
Yeah, this is the last slide actually. We lead to better outcomes, better allocation of resources, etc. And that's pretty much it. I would like to thank everybody involved and the funding bodies, and that's it.
Thank you very much for this very interesting presentation. So there are no questions for the audience, but if there's anything to discuss, we could move to debate lounge 1 that is in the live stream, which is below the button. So I thank all the speakers very much for their contributions, and have a nice day.